All right, so my name is Andy La.
And I'm Nicola Victorov.
We're both from Alberta, so it's our pleasure today to present our research to you. The title of our project is Detecting Enzymatic Cleavage of the Jun B protein.
So I will be introducing the project as well as providing some background information and highlighting our method, and Andy will be sharing with you the results and conclusions of our project. So to provide a quick background on our project, the basic process that we were looking at is apoptosis, or programmed cell death mediated by a group of enzymes called caspases. Now, this is an important biological process in the body as it acts as a regulatory function for cells. And it is known that malfunction of apoptosis can in fact lead to cancer development. So the specific protein that we were looking at is the Jun B protein, and it is known to be highly expressed in some lymphoma cancers. And according to research done at the University of Alberta, it is known that the Jun B protein can in fact be cleaved by caspases. Which leads us to our primary objective, which is to develop an assay for detecting the caspase-mediated cleavage of the Jun B protein in live cells as those cells undergo apoptosis. So the basic strategy that we used for our project is a strategy previously created at the University of Alberta by Dr. Robert Campbell. So we adapted this strategy of using a dimerized red fluorescent protein that expresses a DEVD cleavage site and fluoresces red. What we basically did is we exchanged this DEVD cleavage site for our own Jun B cleavage site, which has been known to be able to be cleaved by caspases. So upon introduction of caspases, this cleavage site would be cleaved, and what we expected to see is that this dimerized red fluorescent protein construct would split up into two DDRFP monomers and no longer show fluorescence. So based on this change in fluorescence, we would be able to examine how caspase cleavage occurs of the Jun B protein. Now, to make sure that it is only caspases cleaving our protein and not another enzyme, what we did is we created a modified Jun B cleavage site, so a Jun B mutant cleavage site. And upon introduction of caspases, what we expected to see is that there would be no decrease in fluorescence. So in this way, we would make sure that, in fact, no other enzyme is cleaving our construct and that it is only caspases. So the significance of our project is the fact that it is a very novel approach, as there is no current technology for monitoring enzymatic cleavage of the Jun B in live cells and in real time. And the applicability of this principle could potentially be that there are many proteins that can be cleaved by caspases. So there are hundreds of proteins that can be cleaved by caspases, and this will definitely aid in research to see how this cleavage occurs. In addition, to take it one step further, our construct can be modified with various cleavage sites. So this would allow to study any type of enzymatic cleavage in cells. So our hypothesis was that the modified construct containing the Jun B cleavage site can be cleaved by the activation of caspases in apoptotic cells, and thus cleavage can be detected based on the changes in fluorescence of this construct. So to introduce the basic methodology of our project, we started off by creating a modified DDRFP construct. And we did this by generating an oligo that is specific to the Jun B cleavage site, following which we inserted this oligo into the DDRFP plasmid. We then harvested this plasmid in E. coli bacteria. And to make sure that this process was in fact successful, that we were able to insert our oligo into the DDRFP plasmid, we purified the DNA from the E. coli bacterial cell. So we took the DNA from the other components of the cell. We ran a restriction enzyme digest, which basically cut our oligo at specific prefabricated sites basically to ensure that our oligo was in fact present, following which we sent our oligo for third-party DNA sequencing to make sure that this was in fact our oligo.
So as you can see, these are results from the gel electrophoresis of our digested DNA. If you look in the first wall, we have our DNA ladder, which is there to act as a marker for the number of base pairs. In the second wall, we have our vector, which does not contain the oligo, so this acts as our negative control. And then in all the other wells are our samples, which include the vector along with the oligo. So what we would expect to see if our oligo had been inserted successfully is that some DNA would migrate down to approximately 750 base pairs, because that's what we determined from the sequencing. So by this process, we would be able to identify the samples which exhibited our insert of interest. So as you can see in samples 10, 11, 13, and 16, all these samples express this DNA at 750 base pairs. So we can conclude that these samples were successfully inserted with our oligo. So to move on from there, we decided to insert this plasmid that contained the oligo into Carpus 299 cells, which are a type of lymphoma cell. So to determine whether this insertion of our plasmid or our transfection of our cells was successful or not, we conducted fluorescence microscopy. So on the left side of our figure, you can see we have our bright field images. So those are present just to show that the cells are in fact there, as not just an empty plate. On the right side, we have our fluorescence images. So as you can see, our vector shows no fluorescence, which is what we'd expect because it does not contain the plasmid. However, our DVD, our positive control, our JUNB, and our JUNB mutant constructs all exhibit this or express this red fluorescence. So this is a very significant result because it not only suggests that the transfection of our cells was successful, it also demonstrates the adaptability of this construct. So even after the DVD linker had been changed to JUNB linker and then changed again to JUNB mutant, all these constructs still express this fluorescence. So it demonstrates the ability for this technique to be used in other methods as well. So progressing on from there, we decided to test for cleavage in our cells. So to do so, we treated our cells with an apoptotic agent, staurosporin, which would activate caspases and therefore induce cleavage. So the test that we did to confirm our results was a Western blot. So on the left, we have our quick exposure. So at the top, you can see we have a PARP protein. So that PARP protein acts as a positive control because PARP is known to be cleaved by caspases. So therefore, if PARP is cleaved, that means that caspases are present, and therefore our construct should also be cleaved as well. So if you note, PARP occurs at approximately 130 kilodaltons prior to treatment with staurosporin. However, after it is treated, it is cleaved to approximately 95 kilodaltons. So this suggests that caspases were in fact activated, and therefore our construct should also be cleaved as well. So looking at the DVD, JUNB, and JUNB mutant constructs, they are expressed at approximately 55 kilodaltons and at a rather high intensity. However, after treatment with the apoptotic agent, you see that this intensity in the band decreases quite dramatically. So that suggests that there was degradation of this protein as well as potential cleavage of the protein. So what we would expect to see if this cleavage had in fact been caused by caspases is that our construct would be cleaved in half. So if that would have happened, we would expect to see a cleavage band that occurs at approximately half the molecular weight of the original weight. So being 55 kilodaltons originally, we'd expect a band of around 27 or 28 kilodaltons. So looking at the longer exposure, we do see these two bands in our DVD and our JUNB constructs that have been treated with the apoptotic agent. And you can also note that the JUNB mutant does not express this band. So that's a great result because we did in fact modify the JUNB mutant so that it would not be cleaved by caspases. So therefore it should not express that cleavage band. However, we do have to note that the JUNB mutant in the quicker exposure does decrease in intensity, so that suggests that there is degradation that was aside from caspase cleavage. So that's something that we would want to look into for further research to see what other potential degradation processes may be involved as well. So the main conclusion that we can draw from our research is that both the Jun B and the Jun B mutant constructs express this red fluorescence in live cells. So this is a really significant finding because it demonstrates the adaptability of our construct, the ability of the construct to be, or this technique to be modified and to be used in various other situations as well. As well, our results suggest that cleavage of our constructs did occur in apoptotic lymphoma cells. However, it's slightly inconclusive as to what extent this cleavage is due to, during apoptosis, is actually due to caspases, though we are quite confident that the majority of it is due to caspases based on the design of our procedure. So as for future work, we would obviously want to repeat experiments so we could learn more about potential degradation processes aside from this caspase cleavage that might be occurring. As well, we would want to examine the cleavage of Jun B with fluorescence microscopy as well as Western blot tests in real time, so by doing a time course. So this would allow us to monitor cleavage of our proteins during treatment with the apoptotic agent at different intervals. So perhaps by the fluorescence microscopy, we'd be able to monitor the change in fluorescence at different intervals, while the Western blot would allow us to know what's happening on a molecular level at these different intervals. So this would really give us a better understanding of what's happening with our construct. So with further development of our technique, it could potentially contribute to other areas of research. So mainly these two areas of basic research as well as applied research. So firstly, this technique could be used to study cleavage in various other proteins. So in fact, hundreds of proteins are cleaved by caspases. So this technique could be used to examine cleavage in all these other types of proteins. As well, since caspases play such an essential role in apoptosis, this technique would be a good way to improve our understanding of this process. And since this process plays a large role in biological function, it's important to learn more about apoptosis. Furthermore, this technique is not limited only to caspase cleavage, but could be used for any type of enzymatic cleavage with further development. So it really shows the diversity and the widespread applicability of this method. As well, in principle, this technique could also aid in cancer treatment as well as drug development. For example, our red fluorescent approach would allow us to monitor apoptosis in cancer cells as they have been treated with new therapeutic drugs. So with this change in fluorescence, we would be able to know whether these cancer cells are being killed or not. So with that change in fluorescence, we could also determine the efficacy of a drug, which is also quite important. Inversely, we could use this technique in normal cells or healthy cells to monitor the effect of drugs on these healthy cells. So in that sense, with the change in fluorescence, we could determine how normal cells are affected and then determine the potential harms of a drug. Furthermore, this technique is not just limited to cancer cells. It could potentially be used in other types of cells. So in fact, both Nick and I were already approached by professors at the University of Alberta, one of which who is studying diabetes, and she's interested in utilizing this technique in her islet cells to see if apoptosis is taking place there. So really this applicability of our construct, of this technique, is really widespread.
So we would like to extend our gratitude to Dr. Robert Ingham and his research group at the University of Alberta, also for Dr. Robert Campbell and Spencer Alford for providing us with our initial construct, and our biology teacher at Old Scona Academic, Mrs. L. Broman.
As well as special thanks go out to Sanofi BioGENEius Challenge Canada, as well as you lovely judges for listening to our presentation, as well as the funding organizations who helped us with our research. Thank you.
Thank you very much.
Thank you. Wonderful. Are you feeling relieved?
A little bit. I thought it went pretty well.
The tough part is still to come. Okay. We have some real tough judges here, I can tell, but they're very
that's
nice.
fine. All right. Yeah, so if
Questions?
you have any questions.
Any questions? Yeah.
I'm sure you're eager to answer them.
For sure. Mm-hmm.
Questions? I'll just get started. In terms of time, give me an idea how much time this has taken. I've seen your names in all the media from Alberta after you won the locals and all that stuff. How does that feel?
It was really great.
Yeah.
Sort of a new feeling, you know, sort of like a celebrity in
Yeah.
Edmonton. But especially our radio talk on CBC was really interesting. That was a really interesting experience going into the live radio broadcast, sort of seeing how that takes place. Yeah, this entire experience has really opened us up to a lot of different opportunities.
Also, it was very interesting how just after the regional competition that we were actually approached by this professor who might actually want to use our work, which really surprised us, is that people will actually be interested in possibly using our work.
It, you know, it makes us feel good about our research.
Definitely.
Jim.
Yeah, so what I find interesting at the beginning is how did you decide to select June B as the first target for developing this method? Because as you said at the end, it's very applicable to studying a number of systems in apoptosis. So what led you to study the June B system, and then how did you proceed from there?
Sure. So initially this project was, the idea was proposed by our mentor because we had first heard of the Sanofi BioGENeius Challenge Canada at school, and we were hoping to pursue this project. So we went to him in hope of being mentored by him. So he gave us this idea for the project, and of course we accepted. So the idea behind the June B protein is mainly because his lab in general is very interested in this protein. So based on his work, he's been wanting to look at this June B protein, but he hasn't had the time, or I guess the bodies in his lab, to look at the protein in depth. So we've been, that's sort of where we came in.
And do you think that your work will lead to a publication? Have you talked about that with your mentor?
We have not discussed it in detail with our mentor, though we believe we could possibly include it in a publication sometime in the future.
What about a patent?
That is something we haven't exactly discussed yet, because of course we're still in the works of, we have to develop this technique. It's not completely worthy of being like patented quite yet because of some of the issues we had with the other degradation processes that may be occurring. So once we fix those up, then of course there's a chance of being able to patent it. Yeah, for sure.
Ron?
So with some of the techniques that you use, could you just give us a sense you were able to do all of those things yourself, run the DNA gels after cleavage and the Western blots? And what antibody did you use for the Western blot?
At the beginning of our project, as both Andy and I were new to the laboratory setting, our mentor did help us in the sense that he provided us with guidance just to be able to do certain experiments. Though as time progressed, Andy and I became more independent of him, and we were actually able to do experiments by ourselves, but just under his supervision. As for the antibodies that we used on our Western blot, we used a anti-rabbit RFP, and a goat anti-rabbit, I believe. Yes.
Nice question.
John?
Mine will be a little bit different, I think.
Sure.
Can you see a use for this in promoting apoptosis?
Promoting apoptosis in what sense?
Well, maybe in plant diseases. So where I'm thinking is that if we can find a system that, if we have a fungus that we want to get rid of.
Sure, for sure. This technique would be able to be used to identify sort of substances that cause apoptosis. So in that sense, yeah, you would be able to identify what sorts of things would be able to kill cells in other environments as well.
So you could use it as a fungicide screen.
Potentially.
Potentially,
with further development, yes.
Interesting.
Marshall?
Just to follow up to Dr. Perlman's question about the antibodies, could you quickly outline what the use of the antibodies you used in your Western blot are? How do they work, and what do they have to do with the actual Western blot?
So let's just go back to that slide. Oh, okay. Yeah, so We had our primary antibody, which was the rabbit anti-RFP. So we had that to detect for the fluorescent proteins that were part of our construct. And then afterwards, we had the goat anti-rabbit RFP that would detect for our primary antibody. So that would allow us to, that provided the color which we would see on the Western blot.
Okay, if there are no other questions, thank you very much for coming, keeping up the level of energy till the end of the day, helping us to keep the energy right up to the level of the day, and keep up the good work. And I hope the media doesn't hound you again all over the place.
Yeah, thanks a lot. Thank you very much.
Okay.
Great pleasure.